Good morning, class. I am posting this video to supplement my announcement that you will find on Blackboard. I hope you had a great week last week. I know that we did not get to meet in person for the class, so I have been very excited for our class this week as we had a pretty fun experiment planned. However, unfortunately, this is how my day has started today. So I have a flat tire on my vehicle and there is no way that I can make it to campus on time for our class. Therefore, what we will be doing today is that we will still do the same lab, but we will do the lab in an online format. While it's not ideal, I know that we can make it work. So here is what I was hoping to cover during this virtual session that we have together. First, I want to start with a few words about last week as some of my students missed some of the assignments. Then I want to talk about this lab. We will go through the same stuff that we would look in a class, see how the lab's hands-on portion would have been completed, and finally we will work through together the worksheet that you will be submitting for this lab. If you submitted your assignments last week, and these included the virtual egg lab, which was in form of a quiz on Blackboard, the Chapter 2 quiz, and Exam 2. Thank you so much for your excellent work. You are the best. If you were unable to get some or all of these sent in, please note that as usual, I am happy to accept late submissions. If you need any of these reopened for you, please send me a message on Blackboard detailing which assignment it is that you need me to reopen for you. I will open these as soon as possible and make sure that you have sufficient time to do these. Especially the exam is worth so many points that you do not want to miss out on those. So let's have a look at what we want to know about the theory for this week's lab. And as usual, we have designed this lab to align with what we discuss in the lecture material for this week. So here is what we will be looking at this lab. This lab is all about enzymes. And enzymes are biological agents that assist in chemical reactions. And as you remember from the lecture and the earlier chapters from this week, chemical reactions were everywhere. After all, as we saw, all life was based on chemistry. How they assist can be varied. For example, they can make the chemical reaction happen at a different temperature than normal, or make it more complete, or somehow more effective. There are many ways. These are just some. And catalysts are a type of enzyme. Not all enzymes are catalysts. But catalysts help with speeding up chemical reactions, so making them happen quicker. And in our experiment, the catalyst would have been liver solution. And why liver, you may ask? I will talk about that in just a few moments, but first, let's have a look of a chemical reaction that we will be looking at in this lab. So here we have it. So in this lab, we will have hydrogen peroxide. So you may have wondered that where have we seen hydrogen peroxide before? And as you may have guessed, it's often used when we want to disinfect cuts and so on. It is because it kills bacteria and other organisms. Your body will also produce hydrogen peroxide as a byproduct of certain metabolic processes. And as it does so, we need to get rid of it because hydrogen peroxide is toxic to the cells in your body. We do so by converting it to water and oxygen. Both of these would be harmless or even needed in your body. So we get this chemical reaction where we are converting the substrate, so hydrogen peroxide, into products, which in this case are water and oxygen. And in order to do this, we have an enzyme in our body that helps to do this quicker, and this is catalase. And this was found, for example, in liver solution. So in this lab, we would use calf liver to speed up this reaction. And then we would monitor the rate of that reaction taking place. So we find ourselves back on this slide. And in this lab, we experiment the impact of two factors on the reaction rate for a reaction that we just saw, where hydrogen peroxide was converted to water and oxygen. We will look at first the impact of altering the amount of enzyme that we add to the reaction. So this will be how many drops of enzyme we put in it. And then we will look at how pH will impact this reaction rate. So we find ourselves looking at this reaction, and what is interesting about this reaction is that as hydrogen peroxide is converted into water and oxygen, this water and oxygen will take a greater volume than the initial hydrogen peroxide. So if we do this in a closed test tube, we will see that the pressure inside that tube will keep increasing as this reaction progresses. And it is this pressure that we would be measuring in this lab by using our equipment. So essentially, the pressure in the test tube will tell us how fast the reaction is proceeding. And it is this pressure inside the test tube that we would be measuring, because it's going to tell us the rate at which the reaction proceeds. So to look at the concentration of the enzyme, we will put a different number of drops of enzyme solution, so cow liver, to the test tube. Let's see what kind of results we will be getting. Now, before we move any further, you should have already done so, or you should pause this video and print the worksheet for this lab from Blackboard. Go to this week's learning module, which will be week 7 metabolism, and click on it to open it. And there you will find the lab titled as Lab Enzyme. Click on it. And if you click on Start Attempt, it will open the lab. It is here that you will find the lab worksheet that you can download and then print. If you need an access to print this worksheet for free, you can do so at our library. Or you can just complete the worksheet electronically on your computer if you prefer. Either way, I'm looking for you to submit the completed worksheet to me at the end of the lab. You will do that as before on Blackboard in this lab's area. So you will find this table on your lab worksheet on page 6, labeled as Table 1. Obviously, since we are not meeting in person today, you will not be able to collect your own group data, so I will provide it to you in this video instead. So here is the data that one of my groups from last night collected. So you can use that for your own notes for this lab. And then what we did is that we had each group to enter their own data on a shared spreadsheet. And this is what it looked like. Most importantly, it gave us class averages. So let's enter those onto our table on our worksheet like this. So that was our first experiment. Now, let's have a look of our second experiment. In this second experiment, we're looking at what kind of an effect will the pH have on the reaction rate. So in other words, how the pressure inside the test tube will be impacted if we use buffers that give us a particular pH for the mixture. You will find a data table for you to complete on page 7 of your lab worksheet, labeled as Table 2. Again, I have shared one of my last night's group's data with you. You can use it as your data. Now let's add the group or class data to this table as well. So by taking an average of all groups data from this class, we were able to produce class data. You can copy it to your own worksheet. Basically, what it tells us is how the pH impacts the catalyzed enzyme, so liver's effect on the reaction rate in this reaction where hydrogen peroxide was converted to water and oxygen. Now we get to page 8 of your worksheet. And this part asks you to look at the results from your table 1. Let's discuss what we see. So the first question asks how many drops of catalase was associated with the lowest rate of enzyme activity as based on the class average. And we will see that the answer to this would be 1 drop of catalase. The second question asks us how many drops of catalase was associated with the highest rate of activity. And here we see that the answer would be 4 drops of catalase. Let's have a look at question 3. Here we are asked to make conclusions about the effect of enzyme concentration on the reaction rate. Well, we see that as more enzyme suspension is added to the test tube on a higher rate, the reaction will present. And we could tell this based on the pressure in the test tube going up. Fourth question asks us to complete group data to the compare group data to the class average data. In this case, they seem pretty similar. There are no large differences. Final question for this part of the lab asks which data set would be more reliable and why? I would suggest that we should use class data for drawing conclusions as it represents findings that are based on a more repeated runs or trials of the same experiment. This helps us to eliminate any variations from the results and get more true findings. Now, let's do the same thing for the page 9 of the worksheet. We will look at the answers to the questions that we have been presented based on our data. So the first question asks us that what pH is associated with the lowest rate of enzyme activity. From the class averages, we see that it would be equally low rate for both pH 3 and pH 12. Neither of these conditions is optimal for the reaction to proceed. Then question 2 asks for the highest rate of enzyme activity. And we see this at the pH 7, which is the neutral pH. On question 3, we should draw some conclusions on this. Well, we see that the rate of reaction would be optimal in neutral pH, so pH 7. And this is indeed the case in human body. Most of the absorption actually happens after the stomach in small intestines. In these parts of the digestive tract, the acidity of the stomach contents is neutralized by pancreatic juice, and now the reaction can start to work optimally. The question 5 asks us about whether we would not want to use, or why we would not want to use the same pipette for transferring different pH buffer solutions. Well, the answer is that by doing so, we would have actually cross-contaminated the samples and the results that we would have gotten would not have been accurate. Finally, let's look at the last question. Here we are asked to reflect on the potential effect of low pH liquids being released into the nature. And this would be problematic for most living organisms as it would alter, for example, their ability to deal with converting hydrogen peroxide to non-harmful water and oxygen. Here we are at the end of our lab. I know it was not quite the same as doing this lab in person, but unfortunately I had no way of making it there in person on time for the class. I hope that you found this video useful as you work through this lab in this virtual format. The final thing that I ask you to do is to make sure that you submit your completed worksheet back to me via Blackboard, as you would normally do at the end of a lab. I will use your submission of the worksheet as an attendance for this lab. Thank you for your understanding, as we have made our best to still give you an experience of a lab despite my car troubles. As always, I am here to help you, so do not hesitate to let me know if you need anything from me. Have a great rest of the week. Bye.